All right, thank you guys for coming. Sorry for the late start. We double booked the room so we couldn't get all set up before you came. So you are here, if you didn't know, to feel like a stewardess announcing your flight. We are going to PhenoGen today. If you do not plan on going to PhenoGen, you are not in the right room. So what I'm plan to do today is do an interactive demonstration of just a few of our tools for you and kind of give you a background on some of the tools and why we like them and why we created them. But we also, at the back of the room, have some flyers, of course, that have the website on it, so you can walk away with something in your hand with the website address on it. And then we also have a feedback form that, of course, we'd love for you to fill out before you leave today to give us an idea of how useful is this to you? Would you likely use it again? What would make it more useful to you? So PhenoGen was first started by Boris Tabakov. He got an R24 grant from NIAAA, which is a resource grant. And he started way back when they were first spotting their own arrays. So he actually created his own arrays back in the day. And we've since evolved through the Affymetrix 3' array to the Affymetrix exon, and now we collect a lot of RNA-seq data. So we've been following this technology all along. And I'll give you a little bit more background of the goals of our project, but first I wanted to introduce a few people. So PhenoGen, the website, the data that we collect is from a group of people working together. So we have people that extract RNA, people that program the website, people that do analyses. And we've got 3 of these people back here today, and they're also going to help us. So as we walk through this, if you run into a problem, if you raise your hand, I have Spencer, Jenny, and Steve in the back that are going to help us work through some of those problems. So that's really the point of this, is not for me to stand up here and preach, but to actually have you guys walk through something so that we can answer questions. So we'll give you an introduction to PhenoGen and what our major goals are with this project, and specifically with that R24 grant. And then we'll go through an interactive demo of a candidate gene approach. So what we're going to do is walk through our browser. So be thinking about your favorite gene, because when we get to that section, I'll ask you to put your favorite gene in there, and we'll work through that gene. I'm going to just do a very, very brief summary of what we call a phenotype approach. So if you actually had phenotype data on some of these mouse or rat models that we're talking about, how you could use our website to go from phenotype all the way to a candidate gene and some biological pathways. And then finally, because I am a statistician, my excitement always goes up when there's downloadable data. So if we're talking about these huge databases, can I download what I want and manipulate it and check it out and see what's going on? And then finally, where we're going in the future. And again, that's a chance for some of the things are where we're thinking we'll go. So if you have suggestions on major additions to add or things like that, we'd really appreciate it. So our goal for the PhenoGen project is to make what we call a transcriptional connectome. So meaning, can we generate a new image of organs as networks of interacting elements? And we're going to use transcription levels to talk about interactions between genes. And so what we're planning on doing is we've already collected full genome sequence and full transcription information using exon arrays on actually we've done brain, liver, and heart, and also brown adipose tissue in one population of rats. And we're also doing our RNA-seq now on brains of these rats and building what we call a hybrid diversity panel that I'll talk a little bit more about later. But we still have in the pipeline to do rat and liver in these strains as well. So why connectome? Most of our research and where this comes out of and where our funding comes from is the Institute for Alcohol Abuse and Addiction. So most of the traits that we're going to talk about, or that we use as demonstration traits in some of these studies, are things that are based on brain activity. So predisposition to alcohol dependence. How does your brain react to alcohol? And how can we predict some of these phenotypes related to alcoholism? And so people have looked at the brain and the connections in the brain in many different ways. They've looked at anatomical or structural brain networks. They've looked at functional brain networks through imaging. And what we're proposing is that we can also use transcriptional information to talk about networks in the brain and overlay that information. So brain, yes. So they are, there is a link on the front page of PhenoGen that has these slides downloadable in a PDF format. So there's quite a few because I wasn't sure how far I'd get, so printing them out became a little bit tiresome. So they are downloadable in PDF format on the PhenoGen site. And so brain, just like any tissue, but is very complex, but brain even more so than something like liver, that you've got lots of brain areas and brain cell types that are talking to each other. And we know that that changes over time and over space. And so how can we dig in and really find out more about this communication at the transcription level? And many of the disorders that we all study are usually not a disorder of a single cell type or a single tissue even, but it's actually how these tissues and how even cells within a tissue communicate. And so we really want to take into account that big picture. And again, one of the methods that we're finding is very helpful into getting to these functional systems and looking at this big picture is graph theory. And so that's what we'll see at the end of this demo of kind of where we're headed. So why study RNA? I just talked about that we think of RNA as another way to identify connections in tissues and cell types and even what's going on within a cell. And one of the reasons that we study RNA is that it's one of the first quantitative links between DNA and the environment. So RNA expression is different in different cells, it's different in different tissues, it's different at different time points. So, but it's all being transcribed from that same DNA backbone. So it's that first chance of actually saying, how does environment interact with your DNA? Hopefully we may get to some of that GWAS gap. I know everybody says they have a way to get at that GWAS gap, but we're hoping transcription is one of those ways. And we can implement things like graph theory and really dig in and look at how does the changing of one gene expression of one gene affect another gene. And we know that the RNA dimension is becoming much more complex than we ever thought it was. I mean, when they first did the Human Genome Project, what was the count? Like 100,000 genes they thought there were in the cell. Well, yeah, it's more like 20,000 protein coding genes, but they can be alternatively spliced to make over 100,000 transcripts. Not to mention all these new non-coding RNAs, so RNAs that don't encode proteins, but are becoming more, we're realizing their biological function in the cell and how important they are, especially when it comes to disease. So how are we going to measure the connection of these transcription levels? We use a co-expression measure. So the theory is that if the magnitude of expression levels of two genes change in a similar pattern over multiple environments, then the two transcripts are involved in similar biological processes. So if they're being turned on or turned up during the aging process and at the same time points, then they're most likely to have similar biological functions. And we're not the first to assume this. Many people have assumed this, and many people have shown some truth to this theory as well. In the case that we're going to talk about in some of our populations, when we talk about different environments, we're actually talking about different DNA backgrounds. So it's a little bit different of a different definition of environment here. And of course, when we look at multiple environments, there are some caveats, especially when we're talking about genetic background, such as linkage to disequilibrium and cell type mixing proportions. So again, we're talking about taking a whole brain or a whole liver where we know there's lots of different cell types in those. We know that the expression levels are going to be very different in the different types of cells. So some of the correlation patterns we may be seeing are because this sample had more neurons in it, where this sample had more microglia in it. The tool that we most often use, and what I'm going to present on here today, is what's called a recombinant inbred rodent panel. So if you're not familiar with this, what it is they take 2 inbred strains of mice or rats or flies or what have you that have been inbred for generations and differ on some trait of interest or differ on a lot of traits of interest. And they take an F1 population, so they get 1 chromosome from the mother and 1 chromosome from the father, and then they interbreed those for an F2 generations, which is mixing up those genomes on any chromosome. And they're going to mix those up quite a bit, and then they're going to start inbreeding for several generations. And so what this does is gives us a very, very controlled population, because these are basically, these 2 strains, a BXD1 and a BXD2, are like brothers and sisters. They both have 50% of their genetics from their mother and 50% of the genetics from their father. But what combination of alleles they have are different. And the fact that they're inbred is a lot of additional helpful properties for us. So this genetic identity within a strain is retained over generations. So that means I can measure RNA expression in the liver, and somebody else can take a different animal and measure the RNA expression in the brain. And we can accumulate all this data because it's on the same genetic background. Where doing this on a human would be extremely tough. And so many of these populations, this graphic in particular is from the BXD recombinant inbred panel, which is one of the more classic original RI panels that people have been collecting data on over many, many years. And they differ on quite a few things. If any of you are into mouse genetics, a BLAC6 and a DBA mouse differ on a whole lot of traits. A lot of people look at them for anything from alcoholism to metabolic traits to longevity. And they make this inbred property also makes ideal controls for studies of interventions or environments. Because just like you can't treat a person with both the placebo and the drug, in a paradigm like this, you can treat one animal from the strain with the drug and treat the other animal with a placebo and control for that genetic background effect. So the panel that I'm going to talk about and do the demo today is the HXB-BHX recombinant inbred panel. And so they were it's a rat inbred panel, and they were bred from a brown Norway that was a congenic and a spontaneously hypertensive rat. So both of these rats are very common in the laboratory setting to then generate both an HXB and a BHX. And the letters just mean the H, the SHR was the mother, and the B, the BNLX was the father, and vice versa. Currently on this RNA-seq, we have brain and males and liver, heart, and breast on the parental strains, and we're working through the recombinant inbred strains. But in addition to these rats, we also have rather large databases on the Lexis RI panel, which is a recombinant inbred panel of long sleep and short sleep mice that were originally bred for alcohol, the susceptibility to the hypnotic effects of alcohol. I'm being very careful to not say tolerance or sensitivity, because it's a mixture of the two. But basically, you give them a dose of alcohol, and one will pass out and stay asleep for longer than the other one. So the short sleep versus long sleep. But they, too, differ on a whole lot of other traits. The BXD panel we talked about, and then also a large panel of just regular inbred mice that get used in studies all over the university. So let's jump right into that candidate gene sample. So is everybody logged on and ready to go? You don't need to register. That's the nice thing about this particular tool. So we're going to go over a genome view and talk about some genome details that are on this interactive viewer, and then we'll talk about some of the transcriptome information, and then get down to what we call a selected feature and go into some detail there. So from the home page, it should look like this. There's a list of different tools that you can use on our website. Anything in green, you don't have to register to get access to. Anything in blue, you need to register. And we're not preventing people from using the data. Anybody can register. It's just with these, you can save your progress. And you can save certain things about your data if you have this login source. Where all of this, if you don't log in, it's gone. Your exact steps are gone the next day. So we're going to click the Genome Transcriptome Data Browser. And so right here, the first thing it asks you is either a gene identifier or a region. So the tools that I'm going to show you today are based on a browser. So you can either enter a gene symbol. It does have to be a valid gene symbol for the rat. So keep that in mind. Or you can enter an actual region, and it'll just pull up that entire region. So my favorite gene lately is the mu-opioid receptor, which is OPRM1. So I enter that in the gene identification, and we're going to choose rat this time. Most of these tools are also available on the mouse. We just don't have as much data across different tissues in the mouse. Mouse is much more brain-centric. And initial view, so that's just telling you what it's going to pull up first for you to look at. And so if you hit go, it should take you there. And so it could give you an error if it doesn't recognize your gene symbol. Or for me, it's telling me that multiple genes are returned, and so I have to specifically check the one that I want. So in my case, there was 2 genes. They actually physically overlap, and so that's why I was coming up with both of them. And so just making sure that I really want the mu-opioid receptor. And we'll hit go. Was everybody able to get it up and going? Great. So I'm going to take this outer scroll box and take it back up to the top. So right now what we're looking at is a regional image. So it's pulling up the whole region of that gene. And up here on the side are tools that will allow you to, if you push this one, you can, just like on, say, the UC Santa Cruz browser, you can select an area and it'll zoom in on that area. This button you can then scroll right and left. This one will take a picture of whatever you're doing, and then zooming in, zooming out, and reversing to what you were before. Spencer is our computer programmer on this website, and he's done a fabulous job. If you hover over anything, you can see when I hovered over this one, it instantly told me what it did. So look for the clues if the picture isn't obvious. So these are the 3 ensemble transcripts for that gene. And if you hover over any of them, it'll tell you a little bit more detail. And then RefSeq has several genes in this area as well. And then next, for the particular RI panel that most of the data is based on, the 2 parental strains, if you remember, are this BNLX and the SHR. And so what this is showing is all SNPs, or small indels, for those 2 strains. And so why that's important with a recombinant inbred, if you remember, these are the 2 parentals. So any of the children, these are the possible SNPs that they have. And the reason that the BNLX has much, much fewer is, if you remember, it is a congenic, and the BN is a brown Norway, which is the reference genome in the rat. So like the C57 is a reference genome in the mouse, the BN is the reference genome in the rat. So we only detected a few SNPs and small indels for this particular strain. And again, if you hover over any of them, it'll tell you exactly where it's located, and then, sorry, shaking hands, the base that was switched in the exact position. So again, like you can see, oh, well, we have one right in the exon of this gene right here, and could look for more details. The next track down is QTL overlapping regions. So these are just published behavioral or physiological QTLs for the rat. So if you hover over one of them, blood pressure QTL, increased systemic artery blood pressure. So all of these have been published somewhere, and we'll show you a place where you can link to all the details about these QTLs. But these have been archived, I believe, by hand by the rat genome database. And we're simply pulling that data and putting it into our image. So one of the things I'm going to have you do, if yours came up like mine, and the next line is selected feature image, so that means I had put in a gene. And so it has based this next image off of the gene. I actually want to X out of this. So do you see the X over here in the corner? Right over here. We're going to close that down so we can look at some regional summaries. So now I have a list of summaries on the side, that there were 3 ensemble transcripts, how many indels, SNPs and indels, and many of these we can click on it, it'll give us a graphic. So for instance, on the SNPs and indels, we can see that 1 was a deletion and 3 were SNPs. When you come down to the QTL regions, it has grouped the QTLs. So we had 8 QTLs that had to do with bone structure and strength. You see that? And then the table below is a lot more detail on that. And so anything in blue is clickable. So these RGD, or Rat Genome Database IDs, are clickable. If you click them, they'll take you back to the RGD database to give you more detail. And then references for these. So where were they published? How do they know that these are QTLs? And then candidate genes and BQTL regions will link to that region within our browser. So one of the other tools on this track details, just want to show you real quick, and we'll go through a few other examples, that if I hit the edit views and change my view, let's say to brain microarray and RNA-seq, hit the apply button, not only does it change our graphic here, but it also changes the summaries below. So instead of seeing QTLs, we can now get more detail about our exons, probe sets from the exon array. So that is a dynamic part of the website. So I want to take a break before we jump into genes with eQTLs in this region, just to make sure that everybody knows what a QTL is. So very briefly, so a QTL, or a quantitative trait loci, is an area of the genome where polymorphisms are associated with a quantitative trait. So we saw examples of bone density and blood pressure. And so what they do is look at markers, what they called markers back when they didn't have such dense SNP arrays and things like that. At each marker, you get a genotype for your particular strain, and you test at every single marker the association of that specific marker with your phenotype of interest. So in mouse, in rat, and rodent genetics, a lot of times what we'll do is what we call a log plot. So this is basically your test statistic. So a bigger value means there's more association there. And so you look all along the genome here for peaks that are above some statistically significant threshold. And so this peak for us, that was quite high for alcohol consumption in these rats. There's also a big range of genes. These aren't little regions. In humans, you usually see this like a Manhattan plot, where they put the -log10 p-value on the y-axis and then the chromosome position on the x. So that's analogous if you're used to humans. So we've done this has been done many years for physiological and behavioral traits. But we've also, and we and many others for several years now, have also thought of RNA expression as a quantitative trait. So now we don't want to only link to phenotype, but we also want to treat these expression levels as a phenotype and see where in the genome is that expression level being controlled. So just like our graphics before, if we have allele dosage here, so AA, AB, BB, for instance, and then looked at the normalized expression patterns, you would see a dose effect for this particular one and say it's associated with that particular marker. And why we're interested in this, because it gives us a little bit of insight into the biology of what's controlling that expression. So we call them both either a cis or a trans eQTL, and cis is technically not right. You should probably say a local eQTL, which simply means that whatever's controlling the expression of that gene is near that gene. So you can imagine a polymorphism in a transcription factor binding site would cause big differences in expression levels, because if a transcription factor isn't able to bind in one strain but it is the other, then you would see a difference in expression. But you also see what we call a trans eQTL. And so what that means is that the genomic element that's controlling expression of that gene is not near that gene at all. It could be on a different chromosome. It could be farther down on the chromosome. And what some obvious examples to that may be, instead of the SNP being in the transcription factor binding site, the SNP is actually in the transcription factor. So that transcription factor isn't getting expressed, and so then downstream that affects many genes, as here in this bottom sample. So the tool on PhenoGen, since we are in a region type mode, we can see what genes are controlled from this region. So is there any genes that might not physically be located in this region, but its expression is controlled from this region? So maybe you would had a QTL that you want to know about, or maybe your candidate gene is a transcription factor binding transcription factor, so you might want to know what other ones are being controlled from here. So what we have here on the left is a Circos plot. And I'm actually going to pull up an easier image for me to show you. The resolution isn't fantastic, but let me give you a general idea. So it's taken that QTL and actually put it in a circle. And the reason that it's doing that is because this, for instance, is the region that we picked out here on chromosome 1. And it's linking to all of these different probe sets or genes whose expression are controlled from that region. So the line goes from where the region that we're looking at to the physical position of some of those genes. The reason it has 4 layers is we have 4 different tissues in this panel. So blue are the QTLs in the brain, orange or red, the heart, green, liver, and purple, brown adipose tissue. Along the sides, you'll see these numbers. These are Affymetrix probe set IDs, and those are the physical location of those probes. And then the p-values are shown as a histogram. And here it is in table form, the same information. How many people have worked with an exon array before? Nobody? So the annotation level over here, let me give you a brief introduction. The exon array is huge, right? It has 100 or it has 1 million probe sets on it. And so it's probing not only known genes and known transcripts, but it's also probing computationally predicted stuff. So somebody thinks there might be an open reading frame, so let's pop a probe set down in this area. And so what they've done is gone through and ranked some of these annotations for you then. So core annotation means they're pretty sure that's a gene, and they're pretty sure they're hitting a gene. Full means they're, somebody said it might be a gene, so we don't really know. So these have the strongest evidence of annotation. These have a lot less evidence of truly being a gene. So we're going to go ahead and not do the WGCNA tab quite yet. Let's start jumping into transcription a little more. So let's go ahead and change our view again. Select Edit Views and do Transcriptome. How many of you have worked with the UC Santa Cruz browser before? So the tracks here are the same kind of concept of what's on the UC Santa Cruz browser. That a track represents information that we have about in this area. So ensemble genes is a track. RefSeq genes is a different track. Alphametric probe sets are a different track. So if we click Transcriptome and hit Apply View, what the default here is, it's going to show you my RNA-seq reconstructions. So we took RNA-seq data from these tissues and did a genome/transcriptome guided reconstruction. So we basically looked at our data and said, what's being expressed? And can we draw a transcript that matches the expression that we're seeing, also using information about what's already been annotated? So we had brain RNA-seq with poly A, and there's nothing there. So that means that this particular gene didn't show up in our poly A fraction. Liver, there's nothing. Heart, there's nothing. But brain, there is a good transcript in there. So for this particular gene, my mu opioid receptor, I have the strongest evidence of expression in the brain. So this, again, is good when you start off with that candidate gene to see what tissues it's expressed in and what isoforms are expressed in what tissues. So since we only have it in brain, I'm going to go back down to my brain microarray RNA-seq track and get a lot more information in a lot easier view. And so now what this one is showing you is this first track is our Alphametrics exon probe sets. So just like I said, they've got 1 million, so they got a ton out here. And they're color-coded to match their detection above background. So that's what the DABG stands for. So these that are bright green mean they're actually detected. These that are black means no expression was really detected there. And so it seems to make sense. We're getting them, for the most part, on our exons. There's also this tool up here in the corner. Our little wheel is settings for individual tracks. So for this particular one, we could click on that. And if we didn't want to code it by detection of background, we could code it by annotation. So that full core extended designation by Alphametrics or by heritability. So that's another great thing about these RI panels is we can measure things like heritability of transcript expression. So with our RNA-seq data, we can also use that tool to expand it to get what we're familiar with here as almost a histogram of RNA expression. And so what's nice about this layout, some of the reconstruction methods aren't all that fabulous, and sometimes they tell us things that are a little strange. So what this is designed for is for you to go through and visually validate what it's done. So we definitely see expressions, higher expression at the exon levels, or rather than the introns. But the other thing we can detect here are reads that cover splice junctions. So did we get our splice junction and intron exon junctions correct? So if you link over this one, you can see that, yeah, we have 131 read counts that overlap that exon junction. So that's quite a bit of proof that that truly is the correct exon junction. While we're here in the viewer, the other thing we can do is add tracks. So if you click on the Select Edit Views again and hit the View Edit Track List, and we're going to hit this button right here, the plus sign, and add a track. And let's go ahead and add our ensemble protein coding genes. So we click on that row and hit Add Track. And then over here, we're going to go Apply View. And so you can see now that our estimated transcript matches closely with this bottom one, but that the algorithm has extended that 3' area. And you can see from our, let me scroll down a little bit, or from our read counts, that there actually is a lot of evidence for that extended 3' area. We do see a lot of reads coming from that area, even though it's not annotated by ensemble. So now let's actually select our features so we can look at some of the selected feature summary statistics. So we're going to click on this one once I get my hand, sorry. There we go. And so what it does is then opens a panel underneath your region image that is your selected feature image. So you can imagine if my region image had been much bigger and included many genes, then the image underneath it would only include the gene that I selected on. So again, we have a lot of details specifically about this expression in the different tissues for this gene, and how many were above background, how heritable they were, and then finally, where are their eQTLs in the different tissues. And again, anything in blue links out to something. So this will link out to Ensemble. This will link out to NCBI to get it directly from the information from them. And now we can get another Circos plot, except now it's specifically for that gene, and it's linking to each eQTL for that gene. So it's a similar concept. So our gene OPRM1 is physically located here, so all of our lines branch out from there. So you can see that many of these eQTLs are tissue specific. And so you can zoom, you can expand, you can say, oh, I don't want to see everything, I only want to see brain, or I only want to see chromosome 5, 6, and 7. And so those are things that you can change within this image. So before we move on to WGCNA, I want to give you a little background on the WGCNA. And it will be quick. So we talked a lot about in the beginning about connectomes. And that was the main reason for doing this project. It's not to make all these conclusions about a single gene, but also talk about, well, what other genes is this gene co-expressed with? And so for genes that we may not know the biological function of, we can start digging into guilty by association type things. So are they correlated with all genes that have to do with protein binding? Or are they correlated with genes that have more to do with transcription? And so what we want to do is look at networks or co-expression modules rather than individual genes. And we, as a lab, tend to use weighted gene co-expression network analysis, which is just one tool for doing network analyses. It's a fairly simple tool to use and fairly simple to understand. That's why we've implemented it on the website. But there's always a better tool out there somewhere that someone hasn't thought of yet, right? So it doesn't simply just look at those correlations to give me connectivity. So it's not just saying what is it highly connected to, but we're going to actually build what we call a scale-free network. And we won't go into all the details here today, but just know that a scale-free network means that there's only a few highly connected nodes rather than every gene. So highly connected genes rather than every gene being connected with 5 genes. And the reason that we want to assume the scale-free network is because that's what we're seeing in biology. And so what this makes it a very robust network, and we've seen it with metabolites and other traits within the cell. And we also want to take one step further too and make a very robust measure of connectivity. We don't want to pull up things that are just happen to be correlated at that time. So to get to that robust measure of connectivity, we're not only going to test the correlation of gene A with gene B, but we're also going to see if gene A and gene B are correlated with the same other genes. So in a social network type setting, are their friends friends? So if A is also highly connected with C, you would assume that B would also be highly connected with C. And so it's just one more way to really make sure that we're getting a robust measure of connection between any 2 genes. So this is one of the examples that has come up in our research, and I just wanted to show it to demonstrate that it's a medium-sized module that has the connectivity shown as lines in between the nodes, which are genes in this case, to show how correlated they are. And then we can also talk about how connected a gene is within a module, and that has to do with the circle size in this graphic. So this one with the biggest circle has the most connection to other genes in this module. And so many times that may have some biological implications. Might not always have some biological implications, but sometimes it does. And so now, instead of looking at individual genes, we have these groups of genes that are co-expressed. So we still want to make some kind of summary of their expression. So the traditional thing to do is take what we call as an eigengene. And basically, that's just a summary of the expression over the module. So it's the first principal component, or something similar would be as if you just took the mean expression within across genes for each strain. We also, some people use what we call the hub gene, or that one that's got the biggest circle, or one that's most connected. So WGCNA on the website, what we've done through the Transcriptome Browser is actually said we had 2 probe sets or transcript clusters for this particular gene, and one ended up in our blue module, and one ended up in Azure. Learn how to say that correctly. And all the modules are named with colors. That's just part of WGCNA's shtick. And so the bigger the module circle here, the more genes are actually in it. And again, if you scroll over it, it'll tell you exactly how many genes are in the module. And so let's look at, if we click on that Azure, the smaller one, we can see a graphic very similar to what I showed you before on the slides. If we move this slider around, we can dictate what lines it shows based on the correlation between any 2 genes. So in this case, our most connected gene is this one. The IMPDH2 would be our hub gene, and our mu opioid receptor is over here. The legend tells you that the green is positive, red is negative, and the thickness of the line goes along with how correlated that connection is. And then in the bottom, it's in table form. And like most things, yes. So we were mathematically pushing the correlation coefficient representation to a scale-free network. So we're taking that, to give you a little detail, we're taking that correlation coefficient to a power. And the power that we take it to is dependent on what power is needed to see that scale-free property. And so that's how we're getting the connectivity between each gene before we call those modules. So, so I guess the question is, mathematically, it's not hard to do. I mean, if you're just talking pure statistics, it's not hard to do. And the beta parameter usually takes care of that and gives you enough control over the network situation. One of the other variables in the WGCNA is how small do you call your modules. So am I going to call 3 genes a module? Am I going to call 30 genes a module? What's the magic number to be a big enough group of co-expressed genes to be a module? And we've chose to go against the default parameters and say that 5 genes equals a co-expression module, where the default would be 30. And so what we're doing is just representing it. And what we see as a hub, there's not always one strong hub. So you're not always seeing what you may imagine in your head as one controlling everything. A lot of times there may be 2 that are highly connected, or there may be one that stands out. But it's not consistent across modules. And so that aspect of a scale-free network isn't forced. Does that answer your question? So just because you might see it elsewhere, I want to click on the blue too to show you what a really big one looks like. So the blue one has over 1,400 genes, and we obviously can't display that in a meaningful way. So what we've done is taken the top 150, and that's what your little caution arrow is telling you. Oops. In the warning up here is that we're only displaying the most 150. But when you come down to the table, all of the genes are there, and you can actually search for your one of interest. So, for instance, there's my mu-opioid receptor 1. And so it's number 620. So it's probably not driving this network, but is a passenger in this particular co-expression module. How connected is it to the rest of the genes in the group? So it's not an exact science. So some of those that are kind of outside that barely made it into your module are going to have really low connectivity. And so I'd put a lot less confidence into them in interpreting the module that they belong to. The higher ranking your gene is within the module, the more confidence I would put to the genes that are closely related to it. No, please ask questions. Right, so it's that co-expression measure that we've taken and tweaked a little bit to get our scale-free network and to get a robust measure. Is how they're put together. We zoomed through time, and I apologize, I probably talked too much at the beginning. So I'm going to just kind of walk you through some of these last slides. Again, you can get a Circos plot for your eigen gene this time. So you can say, is there a common locus of control for the genes in this module? And so it'll give you another graphic like that. And since it doesn't have an origin, it starts in the middle and directs out to where all those EQTL are. And these little X's, it's a little hard to see on this graphic, are the physical location of the genes within your module. There's also a gene ontology tool that is fun for browsing and getting an idea of what ontology terms are represented in this module. And it's very interactive, and I'd encourage you to play with it at some point, because you can scroll over it and it'll tell you what genes belong to that particular ontology category. You can go up or down in the hierarchical chain if you're familiar with gene ontology, to see where your genes are within that chain. And then we also have some tools that look more about expression across tissues. And again, for instance, this is looking at the correlation of individual probes within a gene and seeing how these correlation patterns change over the different tissues. So that's also very interesting if you're talking about what isoforms are expressed in what tissues. We'll skip the phenotype because I'm already out of time and get to the last couple too. Downloadable files. So that was one of the green boxes at the top that you can click on and download most of the data that you saw there. We are in the process of pushing some to the downloadable part right now, so if you don't see what you want, check back with us in a month. If you still don't see what you want, give us an email, and we'll get you access to the data. But it really is our goal to have both raw and processed data available to users so that you can start anywhere in the process that you want to. So if you're not a bioinformatician and don't want to deal with aligning reads, you can actually take that quantified gene level data from our RNA-seq. But if you do want to say, okay, I want to align this with my fancy new aligner, you can download the raw data. And all of the microarray expression is up there as well. And then if you're a really hardcore coder, all of the code is open source and on GitHub. And a lot of our even intermediate code is online on GitHub that we use to process the data to keep everybody in the loop, and we try to update it with where we are in the process of processing the data. Future direction, so in the end, when this whole R24 is done, we will have what we call a hybrid diversity panel in the rat. So we'll have 30 inbred strains of rat and 30 RI strains. So 60 strains total with 4 samples apiece. So we're talking 240 RNA-seq samples on brain and 240 RNA-seq samples on heart. So we're really excited about this panel. We think it'll give us a lot of insight, not only into protein coding genes, but a lot of these things that are being transcribed and nobody has any idea what they're doing. Because if we can say that they are heritable, that's one step closer to it probably has some biological function. If we can identify a locus that controls the expression of it, again, another step towards deciding what the function of some of those non-coding things are. We work with Katarina Kekras and her group, and they've developed a multi-mirror package in R that shows you, scours several different databases that link microRNAs to messenger RNAs, or protein coding RNAs, and gives you both validated pairs and predicted pairs. And we currently have it on the mouse side of our Transcriptome browser and also on the mouse side of the gene list tools. We're currently working some of the databases for it to be available for rat too. So that'll come up soon. We've been asked before to make these WGCNA so you could upload your own, let's say, RNA-seq data and run some of these WGCNA algorithms. So that's something on our radar. Causal inference, you can tell I wrote these slides because they're all statistic-based. Causal inference meaning not only can we say these two genes are correlated, but can we start talking about their relationship? Is it reactive? Is it causal? Or are they just completely independent? And then a Google Drive integration, meaning can we have your data stored somewhere on your Google Drive and then easily integrate it into our data set, into our tools, and be able to use it that way. So I think that's it. I'm sorry I am 4 minutes over. We will stick around for a little bit to answer questions. There is, if you didn't grab one of the feedback forms, first of all, we want to know if this is even anything that you would use. Second of all, you would use it if you added this. And I would use it if you actually trained me to use more than just the Genome browser. So any of that feedback would be great to us, even if it's no, I'd never use it again. So, thank you guys for your participation. I appreciate it. And, sorry, I can't stop talking ever. A couple of notes that there are, the website has, on the front page, has the link to the PDF for this presentation. And it has some feedback links. So if you'd rather send an email, that's an easy way to send it without remembering my name or anybody else's name. But you can also email me directly if you have the flyer. I'm sorry, you had one more question. Where is it? We just added it today. I got to get a little help. Is it on the overview?
Yeah, if you go back to the